Hello, welcome back everyone. Hello George. Today we are going to discuss what was it? It was experiment. We are going to talk about experiments today. And what's the max right now? 118. All right. I'm very calm. The minute it's like always it went over 100 these days. My goodness. Wait. Let me just find okay, there you go. I found the page. I think this what date is? Oh yeah. So let's start. Does my marker work? We have the marker. Yes. Marker test. Yep, it works. It works. So yep. We will talk about experiments today. Let's start with the pulse chase experiment. Wait. Mic check. Let me just listen to my mic one moment. I want to see if it works. I want to listen to it. If it works. It works, right? Okay, it works. Nice. Thank you. Oh, thanks for checking. Yep, so pulse chase. What is pulse chase? So this one is pulse chase. So a pulse chase experiment. It's used to track the movement of proteins within a cell over time. So it will track the growth over time, alright? So first you do a pulse. What is a pulse? So you put in radio-labeled amino acids, and then you do a chase after. You put in non-labeled ones. So you're trying to see if, let's say, the radio-labeled ones, eraser. If the radio-labeled amino acids will be incorporated or not. So these are your radio-labeled, and when they continue to grow, presumably the chase will now be there. The next ones attached will be the non-radio-labeled. And that's how you check the growth of it, like how fast it incorporates the pulse part versus chase part and all of that. So how do you set it up? First, for example, if you're doing it on pancreatic cells. Oh, by the way, it's nice that the bouncy thingies are now working again. So that's good. I don't know what happened last time, but thank goodness it works now. Yeah. So how do we set up a pulse chase? Well, for pulse, you can use guinea pig pancreatic slices first, for example. And then you put in radio-labeled leucine for about 3 minutes. And then after that, you do the chase. You replace the radio-labeled leucine with unlabeled versions, and then you observe the movement of the labeled proteins over time. And then expected findings, all right? So let's divide that into, let me erase it first. Let's erase this one. Expected findings would be different. So for example, that's such a straight line. Oh my gosh, look at that. Wow, beautiful. It's straighter than a ruler. Okay, so this is 3 minutes, and then you have, let's say, 7 minutes, and then you have 37 minutes, and then you have like 100 minutes, all right? So in the first few minutes, the proteins made would most likely only have the radio-labeled amino acids. So take note, the number of amino acids might be attached might be the same per protein, but it depends. It's the incorporation that you have to check. Yup, it's very, very straight. If you check it here, it's incredibly straight. We don't even need an auto straightener for this. And if it's 7 minutes, maybe the unlabeled one will now be attached, and then at around 37 minutes, maybe it will have 2 of the future amino acids, and then in the 100 minutes, maybe only the non-radio labeled will be incorporated. So that's kind of how you check it. Yeah, that's like the hot to cold. If you search videos about it, you'll see the hot and cold terms being used. So yes. Next would be the protease protection experiment. What is that? It's been a while since I read this part, so we will see. Erase, erase, erase. Erase, erase, erase. Okay, good. So next is protease protection experiment. Experiment. So this one, it's designed to check if a protein is inside a vesicle or it's exposed to the cytosol. What does that mean? Okay, so in cells, let me draw a cell again. This is a cell. And then if you have let's say you have the nucleus with the DNA, and then you have your endoplasmic reticulum somewhere here. That's beautiful. And then you have the mitochondria somewhere here or something like that. Anyway, no, not there. Let's say that's the endoplasmic reticulum, and then you have the Golgi apparatus somewhere around here. Vesicles would be the mini. Let's say you need to transport some proteins from here to there. The car is a vesicle. So now this experiment will check if the protein is inside a vesicle or it's just floating around here around the cytosol. What's a cytosol? It's just the liquidy part here around there inside a cell. Okay. Let's see. So yeah, it's to check if a protein is inside a vesicle or inside the cytosol. And it's commonly used to study protein import into the organelles. Are you in or out? Sandara Park has that song. I will not sing it right now. But, yeah. How to set it up. Golgi apparatus. Yes! Guess which one is a GI. Welcome back, the cipher. Guess which one is the Golgi, which one is the vesicle, which one is the nucleus, and which one is the ER. I think it's very, very clear from this drawing. Thank you very much. How do you set up a protease protection experiment? So you first set it up by lysing. Lyse the cells, because of course you need to check it. Lyse the cells to determine, and then you generate the microsomes. What is that? So microsomes are vesicles with membranes derived from the ER. Okay? And then you can do two things. You can do a protease experiment without detergent, walang Tide, or you can use protease with Tide. Okay, so first, if you use without detergent version, if the protein is inside a vesicle, it will be protected from degradation. And if the protein is exposed to the cytosol, it will be degraded. So let's say we use color of Tide would be red, orange. Let's say aloe. Yes, you labhan mo ang cells. You put in the Tide. The Tide will be in like a rectangle. Yeah. Let's say plus Tide, no Tide. I am not sure, and the focus will be on Tide. Regarding, let's say, regarding the Tide picture, we will use it on the proteins. And I mentioned before, if they are inside a vesicle, let's say, here, I have the drawing already. If they're inside here, then the proteins will be protected. But if the proteins are outside then tide will degrade it. Boom. That's how you figure it out. As for expected results and interpretation, yeah, if they don't get digested, then it's inside the vesicle. If they're outside, then they're just destroyed. How? Why? I'm gonna assume the tide destroys some stuff. They disconnect stuff in their membrane or something. It would disrupt it, and therefore the yeah, if they're protected, maybe it's okay. So proteins, let's say, are connected this way, and then they have the bonds, each one. Maybe that's being destroyed and cut up. Yeah, that's how it works. Now, check. Next would be the co-translational translocation experiment. Okay, let's see. Co-translational translocation. So we are now in our third experiment. First one was pulse chase. The second one is the protease protection experiment. The third one is co-translational translocation experiment. What is that? So it's used to determine whether proteins are translocated into the ER before or after translation. Alright, so this one is checking how the proteins will enter the secretory pathway. Lots of pathway. I will talk about them maybe soon. Let's see. So what did I say earlier? Oh yeah, it's to check whether the proteins are translocated during or after translation. Because translation is the process. Remember the central dogma. DNA to RNA is what? Transcription. RNA to protein, translation. But sometimes, as the protein is being made, it can start to be moved. You can move it to another house, essentially. Lipat bahay. Alright? So, how can we do that? There's an in vitro translation system where you can use radio-labeled amino acids to track the synthesis. And you check two conditions, okay? You check two conditions. A, no microsomes are present during translation, aka the proteins are fully synthesized in the cytoplasm before the microsomes are added, or microsomes are present during translation, aka the proteins are synthesized in the presence of ER-derived microsome. Because if they are present during translation, then that means the movement happened while they're moving. No, wait, I have to say that again. Let's say they're cars. You have two houses, aka two parts of the cell, two organelles. And then this is a car. This is a car. What is this car? This is a window. I don't understand. It should look like a car. It has the body and it has two wheels. What else is it? So yeah, this is the car. Wait, there shouldn't be a car yet. Let's say you have two houses. And you're trying to see if the thing was made. How can I say this? If tapos nang paggawa ng thing ba? If the protein was already finished when it was moved, then you don't need the car. But if you can see the car, then that means the car was used. Did I say that right? Okay, maybe. But anyway, if the microsomes are present during translation, proteins are synthesized in the presence of it. Dab. Alright. In condition A, aka the no microsomes, the N-terminal sequence is like still present because they fail to enter the microsome after translation. What? And then B, let's say, no. We have to table this one. Table. I fear that the car analogy might not be enough. Because this is the protein translocation section. Now you see why the heart rate, even without the... Oh, that's such a straight line. Now you see why the heart rate is higher or around the treadmill range even without... Hello, Lendor. Ohio. Yeah. Like, now you see why you need... even without the treadmill, heart rate is high. Because studying is also a workout of the muscles. The brain is the muscle, and your heart will go doki doki if you don't understand the thing, or if you do. Either way, hi, grab the paper. So now we have condition A and condition B. Condition A, no microsomes, aka no car. Condition B, with microsomes during translation, aka the translation, aka the vesicles. Alright, so in situation A, wow, look at that, so straight. Okay, in situation A, the proteins still have their N-terminal N-terminal signal sequence, okay, because they cannot enter the vesicle if they're already a completed protein. They don't need to enter the vesicle because they're finished. But in condition B, the co-translational, the signal sequence, co-translational, and their signal sequences are cleaved. Therefore, that means they were successfully translocated into the ER. This is the endoplasmic reticulum, okay? Because I think once they enter the ER, at some point, the N-terminal sequence is cleaved. So that means if you do not see the thing, that means it was cleaved. Nice. So again, I will close my eyes and try to explain that. So you have the no microsomes and then you have the with microsomes situation. In the no microsomes, you will essentially still see the N-terminal sequence, N-terminal signal sequence, because it was not able to enter. It was already finished and it doesn't need to enter the vesicle. But then if it is in the right side of the table that I drew here, that means it was able to go through to the ER and and its signal sequence was cleaved. Yes, it looks very squishy. That is the ER. Squishy ER. Oh my goodness, wait. My eyesight just cannot see this very tiny figure. So let's see. Oh, I can draw this one. This will help. I'm a very visual person, so pictures help. Okay, let's see. Cell-free protein synthesis. Let's draw the ribosomes. Yes. Eat more carrots. My eyesight might be roughly around the 300 range each eye. That's why I wear glasses during streams. I just change the glasses depending on the thing. It's the fancy Gojo glasses if it's the treadmill one. Sometime, and then during the game streams, I have my on my right eye I would have the mask, and then during the teaching streams I have the blue Sakura glasses, and then soon I will get the lab gown glasses. No, the goggles. Muha. I just forgot to set it up. I will do that in the next one. Maybe tomorrow, or in whatever time zone is tomorrow. Today was is the carrot eyesight thing actually real? I think if I'm not mistaken, it has something to do with, was it beta carotene or something? Let me 1 second. I want to verify this one actually. That's because it's like the squash and the carrots, right? And they're both orange. What carrots eyesight? Let's ask our best friend. Yep, it was beta carotene. And the carotene. And this is the one that we need. So that's why eat squash, pumpkin. Are those the same stuff? Hi, hello Martins. Welcome to the stream. Yeah, eat squash and pumpkins and carrots so that your eyes will be as pretty as Gojo. Yes, that's an eyeball. That's an eye. Okay, and I will zoom in now on my notes, aka bringing the notebook closer, so if the tracking gets cut off, that's how big the notebook was. Alright, this is the ribosome. That's the mRNA. You have the ribosomal subunits, and then your protein will slowly grow like that, right? This is the protein, and you have the N-terminal signal sequence. Oh yes, good drawing, beautiful, not worms of course. And then you have the completed proteins. They will be squiggly lines, and the N-terminal sequences will be, I'll make them red. Eat carrots. Peko shop has tons. Yes, eat carrots. I don't like carrots though. Vegetables. Yeah, I have to start eating them, but the French fries in KFC is too good. Anyway, that's the protein that finished growing. You add the microsome membranes. So if you add the microsome membranes, there is no removal of signal sequence, aka no transport of protein into the microsome. Next is, if you have the microsomes present, essentially they would end up, there's the co-translational transport of the protein into the microsomes. Hi! Nanigaski! Definitely not carrots. Yes! No to gulay, I'm kidding. Eat gulay. Yeah. Vesicle transport assay. 4th experiment. Let's go. What's the first one earlier? Pulse chase. Pulse chase tracking the growth of proteins, and you can check if it's co-translational or post-translational. Second was the protease protection experiment, where you check if it's the protein was inside the vesicle or inside or outside, aka the cytosol. Third, what's the third one called? What was it? The one with a lot of ribosomes. Something co-translational experiment. Yeah, co-translational translocation. Now you have the 4th, aka the vesicle transport assay. What is it about? Vesicle transport assay. It's used to study how proteins move through the Golgi apparatus, aka to distinguish between cisternal maturation model and vesicular transport model. Oh no. 2 terms that I still need to read again. It will be in a future stream, you'll know. It will be called protein targeting, or the Golgi apparatus one. I haven't made the waiting room yet, because I also have to go through the microscopes and model systems too. There's a lot. That's why suddenly this week we have a ton of science streams. I want to play Palworld, but we will schedule that appropriately. We have to get through quite a bit of things first. Yes. So for the vesicular transport experiments, you have the wild type and mutant cells, okay? Wild type and mutant cells. Wild type cells, aka normal cells, contain an enzyme located in the medial Golgi that modifies glycoprotein. What? AKA it has something. It has, let's make a star. It has the thing. Meanwhile, mutants don't have the star. So, the... Nani? I'll just use the right terms first so we can understand. Wild type cells contain N-acetylglucosamine transferase 1, an enzyme in the medial Golgi that modifies glycoproteins. Okay, cool. It's in the medial Golgi and it modifies glycoproteins. Got it. Now, if you mix the Golgi, aka the medial Golgi from wild type cells is isolated, and you mix it with the mutant cells, if the vesicles transport the glycoproteins between the Golgi, then the wild type star will modify the stuff inside the one that does not have the star. Woof, big words. Yeah, that's why I had to read it 3 times, I think. So again, let's make the drawing a bit bigger. It will help. So we have the wild type, and oh wait, Attack on Titan. What was it? The Eldians and the Marlians. Oh my gosh, yes. Okay, we got it. I think this will help. Right? It's the Eldians and the Marlians. The Marlians. Yeah, the X amount of anime watching should finally help. So let's say the wild type, it has the fluid. It has the spinal fluid of the original founding Titan. Yes, see? If I know, thank goodness I watched Attack on Titan. Although the ending, my heart is broken from it. But if it's gonna help me understand science, then I accept it. Okay. So wild type, if it has the spinal fluid, aka the enzyme, that will change up the glycoproteins. This is the glycoprotein, okay? If it has the thing that will edit the glycoproteins, then that's good. But this mutant, the Marlians, they don't have it. But if you mix the wild type and the mutants, but if you mix it, and if the glycoproteins here in the Marlians are edited, that means the spinal fluid of the Eldians crossed and made it through the Marlians. Awesome. But if it wasn't able to shapeshift the Marlians into Titans, then that means Wall Maria and Wall Cena. What's the other wall? I always... it's 3 of the walls. Then that means the walls are intact. Okay? Hopefully that helps. So let's again... The glycoproteins of the mutant cells will be modified if the transport vesicles carry enzymes or when incubated. Oh, Rose, Maria. Oh, Maria Leonora Teresa. Yeah, not that one. It's Maria, Rose, and Sina. Is that the order? From inside. Wait. Now I want to think. It's which wall is the one inside? Where something and something. So 1, 2, 3. No. I shouldn't write. What if that's the one? I'll remember. Yeah, the order. Anyway, they were all broken at some point. So, right? Or I'm assuming all of them broke. I can't keep track now. All I know is that ending, the kokoro was shattered. when I finished it. But what's more heartbreaking is the ending of Oshinoko manga. I will not spoil it. That one. Meanwhile, the ending of Quintessential Quintuplets. Oh, sorry. Yeah, thank you for bringing us back to the topic of the Eldians and Marleys. So, evidence for... Oh, one last thing. The ending of Quintessential Quintuplets. It's not just heartbreaking. It'll make you angry if it's the wrong... If you rooted for the wrong girl. Alright, next. Wigner's vesicle fusion assay. Oh, vesicle fusion. There's so many techniques. Anyway, it's alright. It's okay. It's okay. Calm down. Calm down. See, look, minimum 78. Minimum heart rate. Why is this higher? Wait, 130. It was during the not push-ups, the jumping jacks stream. I know. I was rooting for the most obvious one, and I lost. Like, my first wrong choice. No, my choice wasn't wrong. Author Sama was wrong. Ha. Okay. Wigner's vesicle fusion assay. It's designed to dissect the steps in vacuolar fusion, separating docking and fusion events. What? Okay. Spelling first. Spelling. Spelling. Alright. Wigner's vesicle fusion assay. My handwriting is much more prettier than this. This is just I'm not used to using this one. Cause like, look, even if I try to make it look pretty, the response is not it's not working out. And we continued Quints. I think I only saw Ola Sheriff insert the salute emote if it wins. I never continued Quints. I think I only saw the first season and headphone. Miku was also nice. I was rooting for Itsuki. Wick, the boogeyman's in science. Yes. Yeah. Miku was also good. Nino was alright. But not the one who won. Anyway. Wigner's vesicle fusion assay. Not John Wick. John Wick's vesicle fusion assay. Hello, Ohio, Kyle. Welcome back to the stream. We are discussing John Wick's vesicle fusion assay. I don't remember in which order I watched the movies. Tito, what is that? What aura? Ah, yes, John Wick, Mr. Wick. The famous line of he's a man of, not the myth, the legend. There was a line, something about conviction. I forgot the line. It was the line describing John Wick. Anyway, first, Wigner's vesicle fusion assay, designed to dissect the sequential steps in vacuolar fusion. The pre-existing snare complex is disassembled by Sec18P and Sec17P using ATP hydrolysis. What? Okay, first, priming, then docking, then fusion. It's step by step. You prime it by adding the snare complex. The pre-existing snare would disassembled by Sec18 and 17. How? With using ATP hydrolysis. Okay, cool. And then LMA1. I kid you not. LMA1. LMA1 stabilizes the activated snares for docking. So that's priming, and then LMA1 would stabilize it and help dock. Rab and John Wick, yes, crossover, and Rab would help dock it. Alright, again, you prime it, and then you help it make it dock, and then you fuse. The V snares and T snares would form a very stable snare complex between the membranes. Why do we want to do this? It's just to check the nature of snare-mediated membrane fusion. Alright? Alright, cool. Okay, the next page I have would be, it's from a study called Rapid Redistribution of Golgi Proteins into the ER in Cells Treated with Prefeldin A Evidence for Membrane Cycling from Golgi to the ER. What? Imagine Rin Penrose saying, what? Okay, so it's Golgi to the ER. Rapid distribution of Golgi proteins. Okay. You're checking the proteins that are going from the Golgi to the ER. Okay, cool. And then they're going there. How? You incubate... You incubate the cells with a certain medicine at different times. What was the Lamar 1 again? Lamar 1. You have to say it that way. Not Lamar. Lamar 1. Not Lamar 0. That's yesterday. Lamar 1 is the new Lamar. So it's a complex that stabilizes snares for docking, aka it makes a thing bark. Yes, that's what Lamar 1 does. It's the new Lamar 1. Previously, it was Lamar 0. Now it's Lamar 1. Imagine saying this to your professor. Hey, professor, I learned from TV today that there's a thing called Lamar 1, which helps another thing dock or park. Yep, that's if you have to take away something from today's lesson, it's Lamar 1. Yes, Lamar 2 and Lamar 3. Lamar 0 is out. Well, that's gonna stick. I'm gonna have to sticky note that. I'm going to sticky note Lamar 1. One moment. Sticky note. Oh my goodness, I only have 3 sticky notes left. Going back to the Golgi to ER. You incubate the cells with the medicine. I look it up and get a flashlight helmet mount. I think try to type, let's see. I will try to Lamar 1 dock snare. Yup. You might find it if you type it this way. Lamar 1 dock the snare. Something like that. Those are the keywords that you need to make the NIH articles show up. Because it's related to the ER. Yeah, Google, we have to use Neurosama as the new search engine. Google is failing us. I'm kidding. Incubate the cells with the Prefeldin A at different times, and then you chase it with the fresh media. And you add the pulse first. Permeabilize with saponins, and then you incubate it with IgG, and then you can check it out with immunofluorescence. Microscopy after. I think, okay, that's fine. That is fine. What's the next one? Sec 16, Sec 23, and Sec 17. Those are different, what's this called? Stuff for the snares, or their snares themselves. Oh my goodness. Okay, this is the plot twist. We actually finished classic experiments, and I can actually, should I do microscopes? You know what? It's either we do microscopes or protein targeting and translocation. Anyone want to do microscopes? Yeah, I'll do microscopes, and then I'll just update in the next waiting room. I'll change that. Womp womp. Okay, it's cool that we finished this one. Where's the microscopes? This page. Now, yes. I don't have a microscope cam. I will draw it. It will be much more beautiful. than an IRL microscope, of course. You'll see straight lines, you'll see circles that are perfectly round. Alright, so now we switch and we are now doing confocal microscopy. Whoa. What's the... Oh, I don't have it. Wait. Do I have a celebration emote? No. Dab will do for now. Dab will do. Yes. It is this, even if it's morning, afternoon, evening, or the extremely late evening / extremely early morning, microscopy will be there. Alright. So what is microscopy? Microscopy, what's the goal? What is the goal of microscopy? It's used to create 3D reconstructions of microscopic structures for better visualization or analysis, aka so you can see small stuff. That's it. You make or no, 1 second. Not just the small stuff. You're trying to recreate the 3D, the small stuff so you can see it better. That one, just to make it clear. So for example, you have a mouse lung. Look at this mouse. That doesn't look like a mouse. Oh no. I think we just merged a cat. I don't know what this is. That's chicken legs. Picasso. Yeah, microscopy is gonna be... what did we draw? No, this is a mouse. This is a mouse around your house or in the subways or etc. This is a very chunky mouse or rat, or the cat ate the mouse. Anyway, this is a mouse lung. Oh, so let's say take out the lungs of the mouse and then you have the lung. The lungs would be around 200 microns thick. So it's very, very C. And using microscopy, you can capture a 3D image of it and you can rotate it. The same as if you're trying to make a 3D model, you can rotate it. But unfortunately, I only have a 2D model right now, so you can't rotate it. Even if I try to turn, the back won't show. So yeah, that's the differences between a 2D versus 3D. But it's okay. The tracking still tries to capture them. Oh, and the book goes with the outfit. Yep. So that's how you're trying to capture the image. Next, Z-stacks. What are Z-stacks? Erase. Z-stacks, they're the functions of 3D imaging. Let's see. They're the functions, the foundations of 3D imaging. Because if you want to do a 3D image, individual 2D slices are captured at different depths. AKA, let's say you have a burger, which looks like a ribosome. Oh, that's a horrible burger. Rat, cat, mouse, hybrid. Who knows? What if it was a cat that ate a mouse that ate a rat? Anyway, so this is a burger. This is the Krabby Patty. Let's say you have a burger and this is the meat. Now it looks like a burger. So it's trying to take pictures. Each you cut it up slowly but surely, like this. The different depths. Alright, you see that one, and then you stack the image into Z stacks. And then that's how you recreate the model of your Krabby Patty. How do we acquire it? Good question. So you place low cheeseburger. Yes, good timing. It is a burger. I think there is something wrong with the base of my burger. No, Karen, that's the burger that you ordered, Karen. Next, how do we let's see. How do we... Oh, yeah, you put it on the base first of your microscope. Oh, no, we have to draw the microscope now. 1 second. This is the microscope. Yes, good. So far, so good. And then you have the base. Nice, very nice. And then the stand. Beautiful. So, it does, yup, it's similar, essentially kind of like that. That's kind of how it works, except the MRI, if the human being is lying down, that's a human. If the human being, it's the manager, I am the manager. Yeah. If this is the human being, you want the MRI of the brain, the way you cut up the human picture, the way you take photos of the human is this way, I think, and right, but or some, it depends maybe on the angle, something like that, but yeah, it's similar to the MRI. Good catch. So similar to how presumably that's how you butcher pals, but I've never done it. Ha! I did not make the pallet disassembly conveyor, and I did not make the butchering knife. So no pals are butchered in this space. Even for the hunt of legendary scam. So to start a 3D reconstruction, you place the sample on the stage. You place the sample over here. Okay, that's the base. You place it there. Take the image at the focal plane, move the sample up and down for different lengths, and then compile the Z-stacks. Finished. You put the sample, you take shots, and then you're finished. So there are issues in 3D microscopy. What is the issue? Well, in conventional wide field microscopy, it captures both in focus and out of focus light. So that's why it's blurred. One more time. Why is wide field blurred? It would capture in focus and out of focus light. So the sharp in focus structures and the blurry out of focus background makes your picture very very huge, very ugly. So how do we solve that? Well, confocal microscopy improves the blur problem by what? By blocking the out of focus light. Begone ugh. So on the out of focus background is gone, and you will only see the pretty sharp stuff. So that's why the ability to filter out the background light allows precise 3D reconstruction. So how does confocal microscopy work? Well, you first, there's like 3 steps. One would be, is the heart rate still working? It is, yeah. There you go. So the way it works is, you have the excitation light. It illuminates a point in the sample. Erase. In the Krabby Patty sample. Let's draw the burger again. No, it's okay. You have the excitation light. I want to draw it, but it might... Okay. I'll draw it. This is the burger. You have the burger. Beautiful burger. And then you have the objective. Oh, wait. First, you have excitation light, right? It goes like this. No, I'll draw it in blue. It will be in blue. Blue color. No, not the eraser. Excuse me. Oh, that's the marker. That's the light. Light going there. And then you put... Let's see. You put the lenses here, the objective lenses. And then your blue light also goes like this. So the sample would emit fluorescence, which is collected by your objective lens. Sample's light would be like this. and then you have the tube lens over here. Look at that, beautiful. The tube lens. Lens. May lens. You have the lens, okay? You have the tube lens, and then your camera is up here. Yeah, you have the camera there. So your excitation light, the blue light, illuminates a specific point in the sample, while the sample emits light that is collected by the objective lens, and the fluorescence is then focused onto a camera. So that's how it works. However, fluorescence occurs at multiple depths, so that's why it's very, very, very blurry. Namu namu namu, like the IOI song. And so that's why we use a pinhole. We use a pinhole over here. One moment, because each light you'll have numerous green stuff hitting it at different parts. But if you have a pinhole, aka a purple one here, then the light would just converge at I would say at one point over here, so it will make it all clearer. That's what how your glasses kind of fix it also. So again, what's the issue with wide field microscopy? It would show both the blurred out of focus and the very focused in focus light. The pinhole solution of 3D of confocal microscopy would fix it, because it would remove, or the out of focus light will be blocked by the pinhole, so only the sharpest image is recorded. And a confocal microscope does not capture the entire sample at once. Instead, it builds an image point by point, okay? Point by point, one by one. Isa-isa. One by one. A laser illuminates a single point. The beam moves across the sample in a grid-like pattern, and each scanned point contributes to the final image. The same as how Eren may woke up all of the sleeping titans one by one, and they trampled over how much of humanity was it? 1/4, 3/4, or something before the ending happened. But that's how the raster scanning works, one by one. Next, laser scanning confocal microscopy is next. So they use lasers because lasers produce a narrow beam, allowing precise focusing, and high power illumination ensures fast signal collection. And lasers can be rapidly scanned. It was 80%. Oof. Guess only the main characters are left. Really? Maybe all of them. Maybe. Hopefully it was all of them, but not all of the fan favorites are left. So yeah, that's how laser works. You produce a narrow beam, and then the high power illumination ensures fast signal collection. Okay, so here's the thing. How do we make sure if the pinhole is still there as we move it? So scanning mirrors actually control the movement. So it's not just... So imagine if we have a titan and we need to control the titan. So now it became Code Geass. Okay? And now it became Code Geass. And then you need something to control the... It's like a mecha. Your power... Not Power Rangers. The Transformers thingy. Yeah, you need a thing to control it. And that's where the scanning mirrors come in. They would adjust the angle of the incoming light, and the laser spot is moved across the sample. So that's how it works. And so far we have a couple of things addressed. The first one, let me erase it. Erase. So far we did, which microscopy was first? Wide field. Wide field is good, but the main issue is what? It captures the blur. It's very blurry because it captures the out of focus light. So now we have the pinhole of the scanning of the confocal microscopy anyway. And then now we're trying to fix the issue with that using the scanning, aka the scanning mirrors, so that you can direct the laser beam. Laser beam? What? That's an attack in Pokemon, right? It's an attack. Hyper beam! Okay, it was hyper beam. Sorry. Going back. Why? Okay, there's a thing called PMT. PMT, aka photomultiplier tubes. Confocal microscopes use PMTs instead of cameras because PMTs are very fast, aka nanoseconds, but it's not as sensitive as the CCD cameras. And the speed makes it very essential for high resolution. So how it works is you have the light beam entering the PMT, light beam hits the photocathode, converts light into electrons, and then they're excited by the high voltage. So here's the thing with PMT. They make dim samples, okay? They're fast but low light efficiency. So that's why you use multiple pinholes and a camera, okay? And then that's why you have the spinning disc confocal microscope, which improves upon laser scanning confocal systems by using multiple pinholes. I never understood Pokemon attacks. Like, why can't someone just be like Machamp? Ooh, I fear that if that's the attack, it won't be a rated G game anymore. Maybe that's why we have fancy attacks called Hyper Beam or Thunderbolt. For example, I assume like you have Thunder Wave, right? Because you use Thunder Wave. You're essentially telling a mouse to paralyze someone. So which one is more child-friendly? Pikachu, paralyze him, versus Pikachu, use Thunder Wave. I think it would. And then if you just command Pikachu to be Zeus, aka use Thunder or Thunderbolt, then that's also an issue. Do not mind the background sounds. Thank you very much. It's just the sounds. It's the air. That is the air. Anyway, for spinning disc confocal microscopes, you use multiple pinholes simultaneously, and then that's multiple points at once. It's very high speed. What's the drawback? If the sample is too thick, indeed, it is just fancy wind. If the sample is too thick, the out of focus light can still interfere with the image, so you can't use laser scanning there. So how do you choose which microscope to use? If you have thin samples or very low magnification, wide field is okay. It's also cheaper. But if it's a bit thicker, let me table this. Table time. Microscope. Here. If you have fixed versus live samples, okay? Fixed versus live. Hi. Thank you, Lendor, for passing by. I just randomly joined. Hello, Mamone. What's the lore and what's the topic about? Oh, this is your first time on stream, Mamone. Hi, I'm Millennium, Isekai'd Mask Knight Empress Scientist. I do science, Pokemon, and Pal World, and randomly just recently we figured out how to use the heart rate monitor, so I've been using that while studying on a treadmill. That was a fun one. That's peak insanity. Like you're doing attack, defense, and stamina all at once. That's what technology is today. And the topic would be microscopes now, because we finished the techniques earlier. For microscopes, we are just summing up the different, oh, and jumping jacks, thank you very much, and shiny hunting. I'm at some point I try to do push-ups, but push-ups might take longer because I think and then I'm like, oh, what if the shiny either despawns, runs away, or kills me while I'm doing the jumping jacks. So, but I think that would be fun. And I'm thinking at some point to do. Remember the shinies being voided one, which could potentially be a 24-hour stream. The highlight video is coming very very soon, either tomorrow or 2 days after tomorrow, aka about 3 days from now. One of those. You know what? I may just decide to release it. Hi, thank you very much. I'll release it very very soon. But that's the shiny hunting video will be released. Oh, I remember. The one that's scheduled tomorrow is the shiny compilation. And then 2 days after that would be the highlight video, which just sums up the whole funny void stream. So that's a fun one. I kept on watching it myself, and I was like, that this will be good. So please watch it when it's there. Anyway, for if you have fixed samples or live samples, imagine who is our guinea pig today. If we have, should we use Aaron? Or no? Yeah, I'll use Aaron. No, I don't want to use Aaron. Let's use Pikachu. If you have a Pikachu, no, it's too sad. No, I'll just use a mouse. Hello, Anzaimao. Welcome to the stream. Just on time to discuss fixed versus live samples. If you have fixed samples, you can use, if it's thin, you can use wide field microscopy, because you can just use it. It's cheaper and easier. But if it's a bit thick, moderate thickness, but for still fixed, then you do laser scanning, because out of focus rejection is much better here. If you have very very thick, like just very thick, aka 100 microns, use 2-photon microscopy. Just depends on how thick your sample is. For live samples, thin cells, again, wide field. Thin, you need the thin that needs Z stacks, so just spinning. If you have thick, it's about 30 to 50 then use laser. Then after that, greater than 100, just use 2-photon. So yeah, sum it up. Wide field microscopy is great for thin samples. Laser scanning microscopy provides sharp images, but it's very slow. Spinning disk is faster but is better for live cells. 2-photon is the best for deep tissue imaging, but it's very expensive and slow, so that's why maybe try to not use it on live. And then, but except if it's so thick. And then super resolution microscopy breaks traditional resolution limits, but is technically complex and slow. Yeah, so that's why. So when is laser scanning better? Laser scanning can be better for dim samples if time is not a limiting factor, because you can increase the dwell time per pixel. You don't need to go fast. And since the PMTs are not very efficient, longer exposure will help compensate for it. So that's why you need time for this one. Why is spinning disk worse? Let me use the wave emote as well. Oof. If we have space for the salute one, I think in the post the light stick is winning. But if we have and then in the future maybe it will be the salute one if we have the opportunity. What's the one? Okay, laser scanning is better if time is not limiting factor, but if it's spinning, It is faster but less flexible. And it's better for dim because when oh no, it's faster. When do you use spinning for dim? If the dim signal is still strong enough, and if the sample is live, spinning is more light efficient. For dim, but fixed, laser, dim, live, spinning, very dim, sensitive ones. How can we remember this? If it's dim, I was gonna say wide field? No, it's laser scanning. Dim is dim fixed. Yeah. Anyway, if it's live, just do spinning disc. Okay, question and answers. Explain the principle of confocal microscopy. Confocal microscopy helps you rebuild a structure in a 3D form so you can see it much better, and then you make ikot-ikot it and you can see the back and front and top and bottom. Nice. What is a Z-stack and how is it used to reconstruct a 3D image? A Z-stack is a layer of a photo. It's technically a photo, and it was one of the slices that was taken, and then you stack them so you can reconstruct a 3D image. That's why it's called Z-stack. And then when do we use lasers in confocal microscopy instead of conventional light source? You use it to make the image more intense, so it's more precise. It's precise. Yeah, precise point. Describe the process of scanning and why is it necessary. To make a grid pattern, compare and contrast laser confocal and spinning disc. Ooh, good question. Laser confocal scans the point. Scans point by point for better optical sectioning, but it's really slow. Spinning disc, meanwhile, uses multiple pinholes, so it's quite fast. So laser is good for thicker specimens, but the spinning disc is better for fast live cell imaging. Oh, I just noticed how many of the bouncy thingies do we have now? 1, 2, 4, 5, 6, 7, 8, 9, 10, 11. Yay! I think the max one we had before was 12 or something. Yeah. Or 14. But anyway. Under what conditions would you use 2 photon instead of confocal? 2 photon, deep tissues. If it's very thick, if the sample is very thick, which imaging technique would be the best? 2 photon, because it's the best. It can use it, like it excites the fluorophores only at the focal point. What problem does out of focus light? Oh, because it will cause blurriness. How does a confocal microscope ensure that the pinhole remains correctly aligned? You use the scanning mirrors. What is the role of the PMT? It directs and amplifies the weak signals. What's the limitation of laser scanning and a possible solution? It's very slow because it's point by point, so it would use spinning disc to make it faster. You need to image the neural connections in a live C. elegans sample. Which microscopy technique would you use and why? Spinning disc because it provides fast imaging with minimal photo bleaching, making it ideal for live samples. Nice. Design an experiment to... Yeah, because spinning disc is very fast. So it's good for live samples. It will capture the wiggly movement. Design an experiment to compare the effectiveness of confocal versus wide field. Prepare thick tissue samples. Image them using both. Compare the quality and the background and the resolution. And we will see that wide field would be a bit more blurry. How would you modify a confocal microscopy setup to improve the imaging field while maintaining optical sectioning? Add more mirrors. Spinning disc. Yeah. Why is confocal not extreme for dim? Because... So laser confocal is not extreme for dim because they lose a lot of photons. It needs photons. So spinning disc is much better for if you want faster. Explain why 2 photon is better for thick. It uses IR, infrared light, which penetrates deeper with less scattering. Okay. If a researcher complains that their confocal images appear blurry, what should we do? Or what issues should they check? Pinhole might not be arranged or focused. What is the trade-off between speed and image resolution in different confocal microscopy techniques? Okay, laser scanning, no, it's good. High resolution but slow. Spinning disk is fast, but it's not very resolutiony. 2-photon is deep imaging, but it's slow and expensive. How might future advances improve confocal microscopy for live imaging? So there are some developments in light sheet microscopy and like speed or depth and etc. So that's what future advancements could do. If you were designing a new imaging system for rapid live cell imaging with minimal phototoxicity, what features would we prioritize? This is the last question. We would prioritize spinning disk confocal, okay? Using the CCD camera, the good camera, and adaptive optics for better resolution and minimize laser exposure to reduce phototoxicity. And with that, ladies and gentlemen, we finished the classic experiments and microscopes. Whoop whoop dab. Let me just put the dab here. But yes, so far I think for now that is all for today. Thank you for coming to the stream and learning. Bye bye.